This method can help address key questions in biology, such as how diverse cell types are generated and organized into higher-order structures with specific functions.
The main advantage of this technique is that it facilitates efficient purification of cells present at low abundance in the Drosophila visual system for transcriptomic and genomic analyses.
On Tuesday of week 6 of the protocol, 45 minutes before the dissections, retrieve 1 vial of papain powder from 4 degrees Celsius and incubate it at room temperature. Next, set aside complete Schneider's medium, or CSM, in an Eppendorf tube at room temperature to add to the papain powder later. 5 to 10 minutes before the dissections, use a syringe to add the room temperature CSM to the vial of papain directly through the cap to a concentration of 100 units per milliliter. To mix, first invert the vial to capture any powder stuck on the inside of the cap, and then pipette the contents up and down. After mixing the reagents, bring the water up to 37 degrees Celsius in a beaker in a water bath. Then, place the vial in the beaker for 30 minutes to activate the papain and make sure that the vial is not floating. While the papain is incubating, retrieve an aliquot of proteolytic enzyme blend from the freezer and incubate it at room temperature. After 30 minutes of activation, incubate the papain at room temperature. On Tuesday of week 6, dissect the staged pupil brains in cold CSM with clean forceps.